was wrong to say I wouldn't change a thing, 'cause in the story of our lives, the best of times through color glass, time rolls on enough to let me move on past, but every little now and then it creeps on back to shape my smile.
Today, Professor Matsuda of Kwansei Gakuin University will give a lecture about the photosynthesis in the ocean. So, Matsuda Sensei, if you're all right, please start.
Yeah, okay. Good morning, everybody. Today I'm gonna talk about photosynthesis in the ocean. And before starting everything, I want to introduce myself. I'm born in this part of Japan, which is really northeast part of Japan. That the city is called Kushiro. And then, actually, this is quite close to Russia. Russia is here, and then I quite sometimes saw the Russian battleship on the horizon from my house, actually. So then I went to the Hokkaido University, where I majored the agricultural chemistry and studied on cold tolerance of physiology of higher plants in the Institute of Low Temperature Science in Hokkaido University. Then after that, I moved to another cold place, Canada, Toronto, where I studied, I actually worked as a postdoctoral fellow for the university called York University in Toronto. The topic was changed to study on carbon dioxide acquisition and carbon dioxide sensing system in freshwater green algae. Then I moved back to Japan in 1997, where I got the laboratory in Kansei Gakuin University. At the time, the laboratory was in Nishinomiya, then it was moved to Sanda here. Now we are studying on CO2 acquisition and CO2 sensing system and other aspects in marine diatoms. Marine diatoms I'm gonna explain in detail in today's talk. Quite interesting living organism. And anyway, I want to move into the contents of the study. Okay, today's topic is divided into 5 parts. The first, what is photosynthesis? And second, what are plants in a wide meaning? And third, what is diatom? And fourth, diatom photosynthesis and carbon dioxide concentrating mechanism. And 5, molecular tools and technologies of diatom. So the beginning topic is what is photosynthesis? So the name of the term of photosynthesis, everybody knows it, but probably not many people knows the details. So we're going to talk about very, very basic details of photosynthesis first. So general scheme of photosynthesis is something as follows. Carbon dioxide meet with the reductant, and light is shed in that place, and then organic carbon is produced, and reductant is oxidized, and water is produced. So in this scheme, carbon dioxide is an electron acceptor, final electron acceptor, and H2A is an electron donor, and CH2O is carbohydrate, right? And then there is roughly two types of photosynthesis. One is oxygen evolving photosynthesis, and the other non-oxygen evolving photosynthesis. Oxygen evolving photosynthesis scheme is changes like that. CO2 plus water. Water is a reductant for the CO2 now. Then light is shed in the system, and organic carbon is produced, and oxygen and water is emitted. In this reaction, the delta G for reaction is plus 686 kilocalorie per mole, which is really energy taking processes, and this energy comes from the sunlight. And this photosynthesis is done by cyanobacteria and eukaryotic algae and higher plants, called carbon reduction reactions, not that reaction. Okay, now we move to the more objective schemes, where these two major reaction in photosynthesis is taking place. This is the cartoon showing the structure of chloroplast. And if we see the chloroplast in the leaf of land plant, we can see this kind of how to say cross-section picture. And this cross-section picture can be how to say simplified like this kind of cartoon. And if we magnify this part, then we can see lots of very big protein complexes reside on the chloroplast thylakoid membrane. By the way, chloroplast structure is something like that. It is surrounded by two membranes, which is called chloroplast envelope, which comprises of outer envelope and inner envelope. And within the inner envelope, the space is called stroma. And in the stroma, there is third membrane structure called thylakoid membrane. On the thylakoid membrane, there are lots of protein complexes. So photochemical reaction and carbon reaction occurs in distinct compartment in the chloroplast. And photochemical reaction, which requires light, this thylakoid membrane system is a place for this reaction. However, carbon reduction process is taking place in stroma, which is this yellowish part, not on this membrane. The basic structure of chlorophyll is something like that. And there is a pretty long hydrocarbon tail in tetrapyrrole structure that is called phytol. And this part is called phytol. Tetrapyrrole. Chlorophyll is a green pigment and tetrapyrrole molecule. This one pentagon-shaped structure is called pyrrole. And there's 4 pyrroles here, which is why it is called tetrapyrrole ring. Or in other words, it is called porphyrin. So probably you know porphyrin well, because everybody also has a porphyrin molecule in the body. Do you know what it is? Can I ask somebody about this? Stanley.
Yes, sir.
Stanley, do you know any porphyrin molecule in your body?
Actually, I don't know, sir.
You don't know. Okay, that is called hem of hemoglobin.
Oh,
You-
hemoglobin.
You know hemoglobin, right?
Yeah, actually,
Yeah.
I
Hemoglobin is protein in your blood, which can absorb oxygen, right? Oxygen and CO2 in a different part, but oxygen binding part of hemoglobin has the same structure as this, except it has iron here instead of magnesium. Okay. Photosystem 2 is protein complex, something shown like this. This is a reaction center. P680 is here, and water oxidizing complex within the thylakoid membrane, which is called thylakoid lumen. And this outer side is called stroma, right? And there is antenna pigment here. Antenna pigment takes light energy, and light energy is passed to the reaction center of the photosystem 2, exciting the P680. P680 pass the electron to phaeophytin, quinone A and quinone B, and plastoquinone get out of the photosystem, take the electron energy from the photosystem 2 into the thylakoid membrane. And then plastoquinone is reduced by this way and compose the plastoquinone pool. Okay, so this is a cycle. X plus CO2 becomes 3PGA, and eventually it's gonna be X again. So this is a circulating thing. So they thought that this process is also a cycle. This assumption is sort of genius. You know, we cannot really assume that way. But anyway, Melvin Calvin and Benson Bashan thought that way. But Melvin Calvin only got the Nobel Prize, which is unfair. Okay, we want to summarize this part, and then probably we're gonna take 5 minutes break. Okay, summary 1. Photosynthesis is composed of 2 major components, photochemical reaction and carbon reducing reaction. Photochemical reaction is a system to convert light quantum energy to chemical redox reaction, right? Carbon reducing reaction is initiated by the Ribisco enzyme, followed by energetically uphill reaction into formation of hexose using NADPH and ATP from photosystem. And NADPH and ATP is produced by photochemical reaction and consumed by carbon reducing reaction. The importance of photochemical reaction is to produce high oxidative state to oxidize water, and to produce high reducing state to synthesize NADPH. In the process of photochemical reaction, about 1,000 times gradient of proton is created across thylakoid membrane. So, you know, lumen pH 5.5 and stroma 8.5. This pH gradient, which is called delta pH, is essential driving force for photophosphorylation of ADP by a function of the thylakoid ATP synthase. Okay, this is summary 1, and we're gonna have 5 minutes, and probably is a sort of very deep question. So the question is that one who has leaf, stem, and root? That is exclusive definition of plants? Probably not. And always greenish color? Probably the answer is no. And all evolve in the same way? Probably no. We're going to talk about this. Okay, in 1980s, we start having the technology of DNA sequencing, and then you know, the evolution biologists started to sequence 16S and 18S ribosomal RNA sequence. then they discovered quite interesting point, that is all living organism is grouped into 3 domains. They used to thought that like this, how to say, kushidango, you don't know kushidango, like this, how to say, kebab. Archaea is older, evolutionary older organisms than bacteria. And then bacteria is, of course, is ancestor of eukaryote. That kind of evolutionary tree was believed previously. However, the DNA sequencing changed this idea completely. Actually, there was some initial cell, which is called progenode, was divided into 2 branches in a quite early time of evolution. One became bacteria and the other became archaea. And then from the branch of archaea, there is a sub-branch which goes to eukaryotes like us. Okay, that one I already shown, the color of pigment in diverse plants and chlorophyll. Chlorophyll also I already explained previously, so I will skip this part. And then carotenoid. All living organisms has a carotenoid, type 1 carotenoid like this. So an essential component of all photosynthesis system to protect chlorophyll from over reduction to form triple form chlorophyll. 3 chlorophyll. That's very dangerous molecule. The ability to harvest light is not very high. Only 20% functions of the about carotenoid as an antenna, but rather than antenna pigment, carotenoid can behave as a safety valve of light energy excitation from the chlorophyll A. And also as a type 1 carotenoid, there is a carotenoid called lutein. And as type 2 carotenoid, this peculiar style of carotenoid, which has the Rn and epoxy in the same molecule, that is called fructoxanthin and pyrimidine. This also has an Rn and keto groups here. This kind of chemical structure carotenoid is quite peculiar carotenoid, which has a very good energy transferring activity from light to chlorophyll A. And neoxanthin is also a type 2 carotenoid, and the diatom type 3 carotenoid. But anyway, the kind of carotenoid is in these organisms, and I'm gonna summarize after that. And there is also the other type of the pigment called phycobilins. Phycobilin is a sort of open link chlorophyll. There's 4 pillars, so quite close to the chlorophyll. Chloroplast structure in the secondary and symbiotic organism is much complicated as compared to the primary and symbiotic organisms, and the chloroplast envelope is 4 actually in the secondary and symbiosis.
Wait, yeah, you can start again, Sensei.
Okay, it's okay. I'm gonna start again. So we just we just learned about the structure of the intracellular structure of the diatom, and then the extracellular structure of diatom is a sort of very interesting one also. Diatom has a cell wall made of biomineralized silicate, which is called fluoride. And shape of the biomineralized silicate, which virtually glass, right now, is something like that. Very diverse, depending upon the species. And very interestingly, those the upper view shape is completely a point or line symmetrical shape, And the surface of frustle is filled with a nanometer order arranged structure called areola. This small dot is called areola, like this. And the overall frustle structure is reproduced by generation to generation and species, meaning that the morphology is imprinted in diatom genome. And this frustules is something like a petri dish or shale. We can see the upper cap and lower cap. So what is the trigger for the size restoration is size itself, of course, but also in the case of nitrogen limitation and low salt and etc. other environmental change would trigger the size restoration processes. Size restoration processes is depend upon the types of the diatoms. The most old type radiocentrics, after they prolified, then they will make the okay, so this reddish part is a big cells which doing the photosynthesis. That is estimated as a diatom. And yellowish one is also a large phytoplankton, but which is not a diatom. And blue is a phycoplankton, mostly composed of the cyanobacteria. And greenish one is a Prochlorococcus, which is also a photosynthetic bacteria close to the phycoplankton. This animation is made by Morphology in MIT in 1997. And as you can see, the reddish part seasonally changes, but in the season, in warm season, in each area, this reddish part makes a very massive patches, right? That means their photosynthesis is quite significant in this area. And this diatom photosynthesis from this analysis was calculated that diatom photosynthesis is probably is responsible up to 20% of annual global production, and at least half of the photosynthetic CO2 fixation is down in the ocean, and half it is down in the land system. So 20% of the annual global CO2 fixation means 25% of annual global oxygen evolution, right? So if you take 5 times of the bleeds, like that, 1 out of 5 came from diatoms photosynthesis. So this is very important factor of the oxygen production system on the Earth too. So, you know, as you can see, in January, the southern hemisphere is warmer, right? Then diatom grows a lot here. In April, then they grow in north part more, like from spring to summer, something like that. Okay, so
okay, I'm gonna copy the topic that have been said before. Okay, you guys could also access the Google Docs, but I'm gonna screen share it.
Okay, go on. So I'll stop sharing my screen.
Wait a minute. Okay, everyone can see?
Yeah, still loading in my screen. Yeah, really.
Well, there are 9 people here, so obviously it's main topic. But before we choose to take SDG number 3 and number 6, so the story that we want to construct is being that when a certain algae or certain photosynthetic organism blooms in certain area in water, let's say, then those bloom could affect a lot of marine biota or anything particularly. Maybe it could cause any toxics and any other effect. We want to tell those story to through this topic actually, but we come up with 7 different topic. It's just, does anyone have any more idea like to complete for the number 8 and number 9, or anyone has an objection for this 7 topic that has been chosen?
Actually, for me, the topic number 3, that is photosynthesis and that relate into climate change, is for SDGs number 13, which is the climate change. Because I don't know, I think it's relate to each other.
Wait. Yeah, can't change. But should we make it like the main topic? I mean, we could explain about the climate change itself, but if we only give it one point here in our presentation, could it be included? How to say?
Guys, which number? Oh, okay. Which number do you want to pick? And then then the one who wants to discuss the same point of SDGs, discuss together. Example like I want to choose the number 14, light below the water, and it's same with Rati. So I with Rati will discuss about the number 14 and the correlations with the photosynthesis, and it's also work for the other.
Okay, Pak.
Yeah, just this. That's my opinion. Let me see. Who is missing?
Oh, Gary.
DB. Okay,
Gary.
wait. I'm trying to call her.
Oh yeah, guys, maybe you can find it first from internet about correlate SDG and or SDG correlate to photosynthesis for this topic. Maybe if you already find it, maybe we can discuss from now if you already find it. Maybe I want to search first, and after that I will discuss in here. Okay, maybe that's all.
Like what you said before, the pollution itself. Maybe it could start to talk about the pollution or also the climate change, because
Yeah.
it could also affect, right?
Yeah, or maybe we can spare it into 2.
Yeah.
Yeah.
Oh. It doesn't have to be algae. No sources of blooming. Pollution or and after
And
that,
divide into two general sources. The first is pollution. The second is climate change. Does that make sense?
yeah, that makes sense.
So we already have 4 topics just like that.
Okay, wait. Change. Assumes. And then this is because the ocean acidification, and this is because the global warming. How about that? This point number 4 is actually divided into 2. Like this is the climate change as source of blooming, as in ocean acidification, and number 5 is as the global warming. Like
Yeah, you can make it like that.
wait. Wait.
Yeah.
Does global warming could affect the increase of photosynthesis itself, or it affect the ocean acidification, then the ocean acidification affect the photosynthesis?
No, actually, I don't know. This is just my opinion. As I know, the global warming is that's increasing the water temperature. So it's just like more comfortable for the algae or
Oh
the
yeah,
organism, yeah, to bloom like that.
to be in certain places where they shouldn't be.
Yeah. And
It
but also
will
the eutrophication itself, the increasing nutrition and also the temperature create the algae bloom, something
Yeah.
like that.
Yeah, I think the point number 4 and number 5 must also describe the mechanism. Like maybe because of ocean acidification, it affect a certain thing, and then because of the certain things, the algae bloom or the organism bloom. And in number 5, they could also explain the mechanism, maybe because of the global warming, the temperature of the ocean rise.
Yeah.
It became habitable to certain algae, and the nutrient also converge on those points. So yeah, that blooms. I think this include the mechanism. How about we make number 6 is the effect of the increasing blooming and the photosynthesis to the death of life below water, and then number 7 would be the same effect of this, but correspondent to the SDGs number 3, the good health and well-being of human, and then number 8 would be the effect on the clean water and sanitation. Is it acceptable?
Yeah, you can try it. Oh, and actually, we maybe should ask, could we use another journal outside from the presentation? Because
Yeah. Okay. I think Professor Bibin here. Excuse me, Professor Bibin.
Okay. May I have?
Yeah, I want to ask a question, sir.
Yes.
It's all right. Is it all right to that we add another information from another journal, so it's not stick to like all of the material in the presentation, but add a little bit of information like that?
Yeah, as long as some related with some relation with what is it? SDG. I think it's still okay. Maybe no, because there's no what is it? No information where some Professor Matsuda. No. Okay, we are still waiting from him. So I think, in my understanding, I think it's okay.
Okay.
As long as it has a relation with the SDG, and then also what is it? I think you already select the Meiosis. May I
Yes.
see what you see? Number 3, 6, and 13. Okay, good health, clean water, climate change. Yeah, of course. Reference, I think, is important. In order you would like to make some statement. It is kind of the recommendation. Okay.
Okay, sir. Thank you for the answer.
Okay. How about hello? Hello.
Yes. Yes, sir.
Once Matsuda Sensei come to your room, you can make some confirmation to him again. Yeah.
Okay.
Okay. Thank you.
Thank you, sir. Okay. That being said, let's just roll with the plan first. Plan S, Masuda Sensei when he came to our room.
Photosynthetic product of diatoms. So to me and Latif 一緒にme and Latif look for diatoms together.
Oh, so
Between-
Katsuda, you choose for the SDG number 14, in below waters.
So yes. Latif can you cooperate, Latif?
Ratih. Oh, Ratih. Yeah. Maybe you can chat Ratih via Zoom, via chat Zoom. So because his mic cannot use full. Maybe you can see in Zoom chat, Katsuda.
Hello.
Yeah.
So what is it? What is your question?
Your theme is photosynthetic.
Can you show me which topic has been chosen?
You can choose any topic. You can choose the same topic, and you can give us as many as examples. as you can
So I
find.
can choose the same topic.
Yes.
Okay.
Hello, group 2.
Yeah.
Yeah, only 7 students. Yeah, 1, 2, 3, 4, 5, 6, 7. So
Yeah.
the others, 2 students.
Yeah, and the first one is Monica, sir. Monica that.
Monica in the hospital, yeah.
Yeah, and the second
where's
is Gaby. Yeah.
Where's the Gaby? Because we will have a presentation directly next.
Oh, maybe I will call him. I will call her later, sir.
Okay, please contact her.
Yeah.
If not, because next someone who cannot join with final presentation, we will give some special task. Yeah, but I cannot announce today. But I will directly later on to contact someone like Monica. Okay, thank you very much.
You're welcome.
Guys, I think it's time for break. 10:20. The presentation session would be at 15:40. I think if you haven't finished your presentation yet, just continue it. But if you have, you could take a break, guys. I'll be here.
Konnichiwa, Monikage Sensei. Konnichiwa. Maybe Sensei wa kikoenai ne.
音
Hello.
が小さいです。
あ
まだ小さいですね。あ、
Okay.
聞こえます。
So now student Monikage Sensei.
Hey.
Student actually now already break time, and but many students still in the breakout room. But maybe it's about yeah. I think I will close the breakout room and they will come to the main room.
So they come back to this room?
Yeah. I will close
Okay.
the all room now.
They are in a breakout room now.
But already finished now. Okay.
Mostly, okay.
Well, I think now I will let Matsuda Sensei and Monikage Sensei to achieve this kind of final presentation. As we already informed that presentation per group, they will have 30 minutes, and then also each group will have 15 minutes for discussion.
Okay, so let's start the seminar by 3 groups from group 1. So please, group 1, start please.
Excuse me, sir. First of all, good afternoon. I'm Timothy, the leader of Group 1. And this afternoon, Group 1 would like to present about the UN Sustainable Development Goal in connection with photosynthesis in marine science. There are a few SDGs that we have chosen to be presented about, and each of us has a different topic to talk. And so, Group 1 consists of An Agung Ole, Harada Ikari, Jovita Larasati Putri, Kawasaki Kai, Made Prasasti Abelia Oksana, Putu Ilandari, Putu Echa Priyaning Aryunisa, Shiro Yamashogo, and me myself, Timothy.
Okay, thank you for the time. On this slide, I will tell about general explanation of photosynthesis in marine. Plants, algae, and cyanobacteria are engaged in oxygenic photosynthesis, which means that they require water and release oxygen. Phytoplankton plays an important role in the process of providing oxygen on marine. Phytoplankton is a large set of a billion of microscopic algae that live in the ocean. These algae plays the same role as plants on terrestrial land. Even tiny phytoplankton release more oxygen into the air than all the forests of the world.
Okay, now I will talk one of the photosynthesis on marine ecosystem, which is algae bloom. Next, algae bloom, the massive photosynthesis which has a bad effect. We can say that photosynthesis can be bad if it happens excessively. And one of the massive growth of organisms that is bad for environment is algae bloom, or we can say harmful algae blooming. Global trends in the occurrence of the toxicity and risk posed by harmful algae blooms to natural system, human health, and coastal economies are purely constrained but are widely thought to be increasing due to climate change and nutrient pollution. Okay, this is the example of a life cycle of Alexandrium as species. This kind of species is also one of aids the successful establishment of bloom populations. Uh, Generally, the dinoflagellate produce a resting stage or a cease during periods of suboptimal growth conditions. This is why harder alga blooming could be happened, which the seeds sink to the bottom and after an obligate of dormancy periods, or they can exist or break open to release vegetative cells that swim to the surface to reseed bloom populations.
I take up. Hi, I'm Shoshima. I talk about ocean acidification. Carbon dioxide dissolved in the ocean becomes H2CO3. In the ocean, H2CO3 is in a state of chemical equilibrium due to the reaction between H plus and dissociated HCO3 minus and CO3 2 minus as atmospheric carbon dioxide increases, so does the amount of carbon dioxide dissolved in seawater, and the reaction proceeds to produce hydrogen ions H plus. Most of the generated H plus is consumed as the reaction proceeds. But some H plus remains intact and CO3 2 minus degrees. As a result, the pH decreases due to the increase in H plus. Various marine organisms such as the phytoplankton and the corals build skeletons and shells of calcium carbonate from seawater that are insoluble in water. However, as ocean acidification progresses and hydrogen ions in seawater increase, it becomes more difficult to form calcium carbonate shells. Thank you.
I think it is muted.
Oh, I'm sorry, sir. So the blooming have effect on a lot of things, which one of them are human health. Mostly it's come through the food and water poisoning, skin contact infection, or either poisoning through inhalation to respiratory system. For the foodborne, there are a lot of poison usually are while eating shellfish, because shellfish are a filter feeder, so they filter a lot of things, which one of them are the blooming of the algae, the diatom, the dinoflagellates, a lot of that. And the poison could be amnesic as a parasite, gives diuretic and then neurotoxic, paralytic, and for the fish, there are ciguatera fish poisoning or polytoxin, and also the waterborne. And for the aerosolized biotoxin, it came from the Carenia brevis, which is brevetoxins, and also Osteopsis CF ovata, which give the respiratory irritation. Other than human health, the blooming could affect also the human well-being, which is could compromise the SDG number 3 for the good health and human well-being. This particular study comes from the Cornwall, United Kingdom, especially on the St. Austell Bay on southwest England. The study came from the 1st January till 31st December 2014, and based on the data from the FSA through 22 culture producer affected. These 22 aquaculture are culturing a lot of shellfish, clam, and organisms like that, and almost all of them are affected by the bloom. They tested positive for the toxin. and it gives a lot of effect in the personal level. Like a lot of like local diving instructors have to cancel all event, and fishermen aquaculture have nothing to catch, no money for them, and then the related industry to all of those infected are also get affected. And then for the personal level, for individual, there are reduction in experiences primarily on the relaxation experiences, stress reduction experience, and also inspirational activities experience. And then on the communities level, Cornwall was iconic for their clan, for their aquaculture, and because of this condition, or because of this phenomenon, Cornwall has suffered greatly because they can serve a lot of their iconic land, their iconic aquaculture product because of this. And so it affect on the local sense of distinctiveness, attachment, also identities for their people.
Then we arrive on the effects of increased photosynthesis and blooming to clean water and sanitation. First, it's create dead zones and hypoxia in the water. Harmful excessive blooms cause massive death of aquatic organisms that include fish, plants, and also decomposes bacteria, made thick and green mark that impacts clear water.
Okay, probable solution. Microalgae for bioethanol.
For the good health is SDG number 3, and then clean water is number 6, and then the climate change would be number 13, sir.
And health, where's the health?
Health is number 3, sir.
Number 3. Oh, okay.
Yes.
Why you choose these topics, the combination of this topic?
After a careful discussion between us, we choose that explaining about the increase, like say, increase in the photosynthesis activities is also the blooming of the organism, like microalgae, like dinoflagellates, that could be pretty interesting. And also we would want to like comprise some SDG point, the effect of the blooming itself that could affect human, not just human, but the environmentally itself. So it could explain pretty much a lot of things. Maybe
Okay.
that's it, sir.
So we're gonna move to the group 2, and please, group 2 start.
Yeah, okay, sir. Thank you. Thank you. Maybe GP will share screen. Okay. The first one, our group with explanation about correlate photosynthesis to 4 sustainable development goals. They are number 7, number 2, number 7, number 14, and number 17. And they are our group. The name is Stanley Novans, and my friend's name Gabriel Aisha. And the next is Yang Yingjian, and the next is Hareru, and the next is Modika, and the next is Baba Kresna, and the next is Puturati. Next, Anggi Hekson of Victoria Manik, and the next is Katsuda Yuki. Next, Gibi. Yeah, the first one is will introduce with Katsuda. Katsuda.
Sorry. Okay.
Yeah.
Can I listen my voice?
Yeah.
Okay.
You can start it.
Next, please. Okay. I think diatoms will help solve that zero hunger SDGs 2 solution. The reason is that diatoms have been eaten a long time ago, so I thought it would be useful for solving this theme. The main photosynthetic product of diatoms is triacyl glycerol. Although it is expected to be used as a biofuel, triacylglycerol is one of the neutral fat and can be used as an edible oil. Neutral fat are stored in the liver, and if you are deficient in sugar during exercise, you can use neutral fat instead to move your body. In other words, neutral fat are the energy to move the body. In addition, neutral fat have the function of keeping body temperature constant. Triglycerol, which is obtained by photosynthesis of diatoms, is considered to be a necessary energy source. Also, in fact, fossil diatoms are sold for food. It can eat diatoms. Moreover, it has also been selected as NASA's space food for two reasons. I think diatom will help solve the SDG2 theme. Next, please.
Yeah, as you can see in the PowerPoint that I already add, I choose I already source and I found some papers, and the name papers is the potential of the atom biotechnology for the application in solar cell, batteries, and electroluminescence device. So I think this paper can correlate to sustainable development goals number 7 because it provide to help to have like solar cell to make electricity source become better and become efficiency. Next, Gaby. Yeah, as you can see in the PowerPoint that diatom will have a frustal that in diatom that frustal can make and can help like in the paper before to insert of as titanium or germanium dioxide into the nanostructure of the diatom frustal that can make and can help in solar cell like sensitized solar cell. And the next is nanostructure battery electrode, and the next is electroluminescence display device. So the point is diatom can help in sustainable development goals number 7 to efficiency to make solar cell, battery anodes, and display device. Next.
In this slide, I will introduce the battery, which utilizes biofuels and photosynthesis mechanism. First, today, many of us use fossil fuels energy, which is essential in our daily life. However, the energy produced by fossil fuel is limited resource, and moreover, it emits CO2, which is known as greenhouse gases. That means we need the more sustainable and clean energy instead of fossil fuel. The idea to solve the matter is using solar energy to energy conversion mechanism of photosynthesis. Sorry. In this slide, I will introduce how to make battery from biofuels such as cyanobacteria. The cyanobacteria oxidize water and produce oxygen when they absorb light. In this reaction, electron is produced and conveyed by this scheme. Then NADP plus is reduced by electron and used for carbon dioxide fixation. Next slide, please. Researchers constructed the battery and had experiment whose anode is cyanobacteria and H and Q and phosphate buffer, while cathode is potassium ferricyanide and phosphate buffer. They exposed battery into natural gas, including CO2, with light and got electric current. In this research, about 18% of glucose inside the cyanobacteria is converted into electricity. However, the biomass energy will still has challenge because their lives have limitation. To use it efficiently, researchers tried to use a silicate membrane and put it on anode surface and irradiated light. As a result, they got same voltage. However, they got less electronic current than using the cyanobacteria itself. When they have much lower, that means they still have challenges. Finally, research on battery using solar energy by using photosynthesis mechanism have been conducted since 1960 and continuing and it's still continuing to reduce greenhouse gas emission and use sustainable clean energy. In this field of research will be more important than before, I think. Next, please.
Okay, so for this one, I'm going to discuss the 17th goal of SDG, which is the last, is partnership for the goals. So as UN says, the point of the 17th point of SDG is to enhance a global partnership, especially in third world countries, where a successful sustainable development agenda requires partnership between governments, the private sector, and civil society. These 17 ambitious goals is gathered into one complexity of this last SDG because we have to work together to make a sustainable world where we can live together. Okay, so for a case that I'm going to tell you about is in Indonesia, where the sectors that can take part in this SDG is all sectors regarding the government or even private sectors. In this government, I take an example of the marine and fisheries government in Indonesia, and also a private sector, which you can see in the slide, where they can work with non-profit organizations such as Carbon Ethics and Sustainable Ocean Alliance. And then from their non-profit organization, they can also take civilians and also societies to come and work with us together to reach the goal of the SDG. So about carbon ethics, it is like an NGO organization that aims to decelerate the climate crisis by caring for our carbon intake and carbon offset. So they grow mangrove trees, they also grow seaweed and coral in regard to saving the blue carbon in our living. And then the next one is about Sustainable Ocean Alliance. So Carbon Ethics is from Indonesia, and that is one of the example of the national NGO that we can collaborate with to achieve the 17th SDG. And then the second one is Sustainable Ocean Alliance. This is a global one, which is made in Georgetown, and then now they have over 165 people all around the world and in all countries, which are making and finding solutions regarding sustainability regarding the ocean. And this can, they are focusing on the ocean, but we can also focus on a lot of things regarding sustainability, such as food and also the climate and everything like that. So in Sustainable Ocean Alliance, they select a lot of people to come into their community to make a solution. And then they also give grants and fundings for people that have solutions regarding sustainability. And I think that in achieving the 17th SDG, we have to work together with NGOs like these all around the world because as SDG says in the UN environment, there are no borders in nationality or in race or in countries to be working together to reach the sustainable goals. Thank you.
Okay, thank you. So any question?
Excuse me, sir. I have a question.
Yeah, please.
For group 2, maybe it's not directly connected to the photosynthesis, but I'm curious actually. Like as we know, coral reef, a lot of coral reef area are actually experiencing a bleaching, and that's because like maybe the temperature rising, so the zooxanthellae are leaving them. But I'm curious, are there any way to maybe culture those algae again, culture the zooxanthellae, and then reintroduce to the recently died or recently bleached coral reef? I think that's my answer, sir. That's my question, sir. Sorry.
Anyone can answer to this?
Okay, thank you for the question, Timothy. So the question, if I could conclude that about if the zooxanthellae or this macroalgae has been out from the polyp of the corals, how to make them back again, something like that?
Yes, more or less.
Okay, so to asking about that is all depends on the environment situations. So when all the environment factors back to normal again, when the zooxanthellae could live in this kind of waters, so it's naturally will back again to the polyp of corals. So in this case, just only happen if the
Baba
and the reefs still have the polyps. If the polyp has been died, there's no place for the zooxanthellae because the zooxanthellae live on the polyps. So there's two points to make the zooxanthellae back again. The environmental conditions should be suited for the zooxanthellae, and the second one, the polyp is still there on the reefs.
So lots of absorption space like a charcoal. Okay, so which is why that might is suitable for the how to say house interior to absorb humidity and to absorb the bad smells and to absorb the bad chemicals. Yeah, but it is a story of the diatom fossil, which is a diatomite. We know to produce that amount of diatom fossil by the culturing, probably we need to culture huge amount. Yeah. Okay, so that's my comment. Maybe so we can finish.
I think so. Thank you very much, everybody.
Yeah.
I will ask Professor Tosensei.
Okay.
Yeah.
Okay, thank you very much for lecture and discussion, Matsuda Sensei and Murekage Sensei. And thank you very much for nice presentation and active discussion. All the lectures have finished today, but the 4 presentations remain next Sunday. So good luck on your presentations, and I'm very looking forward to watching your presentation next Saturday. Okay, let's close today's program. See you next Saturday, and sampai jumpa lagi. So bye-bye.
Bye-bye.
Dadah.
Thank you, sir.
Sampai jumpa, sir.
Thank you, sir.
Yes.
Thank you, sir.
Bye.
Thank you.
lit this town like a fire in a blaze, gotta burn it down. Can't be afraid to leave this out. We got this far, don't know how. I see danger in your eyes. They know we'll burn down the night. Coming just like the sunrise. You know that we gonna light it up tonight. We gonna light it up tonight. We gonna light it up just like the sunrise. We gonna light it up. Just like a spark that spreads